Myself, Mr. H.T. Mate, Department of Botany, RBNB College, Sri Rampur. So today we are going to study the T.Y.B.Sc Botany, paper first BO 331, that is the cryptogamic botany, and from cryptogamic botany, topic number 5, that is the life cycle of the Rhizopus. So first the classification. In the classification, division is Eumycota, and the reason is the presence of a cell wall throughout the vegetative phase. Subdivision Zygomycotina, asexual reproduction by non-motile spores, and perfect stage spores are the zygospore. That is the class Zygomycetes. Morphologically similar gametangia are found in the Rhizopus. Order Mucorales. The reasons: number 1, that is the mostly they are saprophytic in mode of nutrition. Second, asexual reproduction by non-motile aplanospores or simply the spores. Then family Mucoraceae, that is the sporangia with many spores, and at the base there is the columella in the sporangia, and genus Rhizospore. Now this is all about the classification, and next point, that is the Thalassa structure. Now Thalassa structure, this is branched, white in color. It is a tubular, it is a stenocytic, it is a multinucleated. Vacuole, endoplasmic reticulum, glycogen, which is the result of food material found in the Rhizopus. Oil drops are there. This is the diagram of young mycelium of Rhizopus. Now it shows branched white cottony structure in appearance. It is branched, it is white, it is tubular, number of nuclei are there, there is no septa. Cross walls are not found in mycelium of the Rhizopus. So such a type of mycelium is called as the cenocytic mycelium. So the type of mycelium is a cenocytic, that means there is no septa, there is no cross wall. And in a tube-like structure, in a tube-like mycelium, number of nuclei are simply suspended in the cytoplasm. Now number of cell organelles are there, and food material, that is the glycogen and oil drop are the food material. And cell organelles, for example, endoplasmic reticulum is there. So such type of a number of cell organelles are there, number of nuclei are there, and a number of vacuoles are also there. So this is the structure when mycelium is young. But at the time of reproduction, this mycelium is get divided, it is get differentiated into 3 different types of the structures as follows. Now the structure of a mycelium at the time of reproduction. So this is the stolon, it is branched, it is white in color. So these are the rhizoids, they are branched, they are also white in color. This is the sporangiophore. which is elongated tube-like structure. These are the asexual spores. So this is the diagrammatic representation of the one species, that is the Rhizopus stolonifera. The whole mycelium at the time of reproduction, at the time of sexual reproduction, it is getting differentiated into the three different parts. The lower parts, which is in the substratum, is called as the rhizoid. This rhizoid, they penetrate in the substratum and absorb the ready-made food material from the substratum and also absorb the water. They are branched, they are also white in color, and they are also cenocytic in nature. That means they do not have the cross wall. So this is about the rhizoid. Now about this, that is on the substratum. On the substratum, another type of a branch which is called as the stolon, they creeps over the substratum. They are also white in color, they are cenocytic, they are branched, and they are cottony in color. Now they are cenocytic in nature, they are branched, and they develop the tufts. And at the site of a tuft, at the site of a node, from lower side they develop the rhizoid, and from upper side they develop the sporangiophore. So this is about the stolon, which is the branched cenocytic in nature. And above this there are the aerial structures, and they are called as the sporangiophore. These sporangiophores are elongated tube-like hypa. They have number of nuclei. At their tip there is the sporangium. Sporangium is somewhat circular in outline. And at the base of this sporangium, there is the columella. And above the columella, that means in the sporangium, there are number of the spores are developed. These spores are asexual spores. That means when they get germinate, they develop into a new mycelium of the Rhizopus stolonifera. So the mycelium at the time of reproduction is get developed into rhizoid, stolon, and sporangiophore, which has the sporangium. So this is about the structure, Thalassa structure of the Rhizopus stolonifera. Now next point, that is the reproduction. Now Rhizopus reproduces by two different types of the methods. Number one, that is the asexual reproduction. It is again of the two different types. And second, that is the sexual reproduction. So first, that is the asexual reproduction, which takes place by the formation of the asexual spores, which is called as the aplanospores, or simply the spores. So at initial stages of the development, a small circular structure is formed, which is called as the vesicle. Now this is the stolon. This is the columella. Sporangiferous layer. This is columella zone. Sporangiophore. Now this is about the asexual reproduction which takes place with the help of aplanospores or the spores. Now at the initial stages of the development, there is the stolon, which is the branched sinusotic. It develops some elongated tube-like structures. They are rod-like structures, or they are called as the hypa, and these hypa are called as the sporangiophore. Now number of nuclei which are in the stolon and cytoplasm get migrated into this sporangiophore. Now this sporangiophore at its tip develops a small circular structure which is called as the vesicle. Now all the content, that means the number of nuclei, cytoplasm and other content which is here in the sporangiophore is get migrated into the vesicle. After that, vesicle becomes large in size and it is get divided into the two parts. The part which is at the base is called as the columella, which is less denser, and above this this columella is called as the columella zone, and above this there is the sporangiferous zone. Now this sporangiferous zone with a number of nuclei and cytoplasm get metamorphosed into small circular structures. That means in each circular structure there are number of nuclei, cytoplasm, etc. is there, and now these circular structures are called as the spores. Now whole this structure is called as the sporangium, in which large number of the spores, which are asexual spores, are there. And at the base there is the columella, and below the columella there is the sporangium. There is the sporangiophore. Now when these spores becomes mature, the wall of this sporangium dries up and all the spores are get dispersed with the help of wind. And when conditions are favorable, all of these spores, asexual spores, get germinate into a new mycelium by developing a germ tube. So in this way new mycelium is get formed. For example, this is takes place in a Rhizopus stolonifere. Now second type, that is the Climato-spores. Second type of asexual reproduction which takes place with the help of Climato-spores. Climato-spores, simply called as the Climato-spores. These are the Climato-spores. They are thick-walled. So when conditions are unfavorable, the sinusotic mycelium becomes a cellular, and some cells, they secretes a thick wall, stores very large amount of the food material and slightly large as compared to the size of the mycelium, and these newly formed cells are called as the Climato-spores. and when conditions are favorable, each climatospore get germinate into a new mycelium of the Rhizopus. So this type of climatospores form when conditions are unfavorable. Now next point, that is the sexual reproduction. Now next point, that is the sexual reproduction in the Rhizopus. Now at the time of sexual reproduction, two opposite strain mycelia or hypa come close to each other, and now they are called as the zygophore. They are called as the zygophore. From these, one is of the plus strain and another is of the minus strain. They come close to each other because of the sexual hormones secreted by both the zygophore. Now at their contact, they develop a small tube-like structure. So this is the tube-like structure. They are called as the copulating branch or the copulation branches or conjugating branches. Now these copulating branches are get developed into a large structure, and now they are called as the gametangia. So from both sides, there are the two opposite strain gametangia are there, and both are multinucleate. Now both these gametangia, at their tip, they cuts a small cell from both the side, and these small cells are called as the gametangia. That means there is two types of the gametangia. One is of the plus strain and another is of the minus strain. Now they are denser. That means they stores very large amount of the food material and very large number of the nuclei and other cell organelles, and the part which is behind is called as the suspensor. So from both the side there are the two suspensor. They are vacuolated and they are less sensor. Now karyogamy takes place in between these two. Before this, the wall in between their contact, there develops a small pore, and through this pore all the nuclei fuse with each other to form a diploid nuclei, which is called as the karyogamy, and a new cell is formed. That cell is called as the fusion cell, in which all the nuclei are diploid in nature. Now this fusion cell enlarges in size and becomes circular, and now this circular cell is called as the zygospore, which has the two layer. Outer layer, which is thick and hard, is called as the exine, while inner delicate layer is called as the intine. It is called as the zygospore, and all the nuclei are diploid. Now this is zygospore. Germination of the zygospore takes place. Now this exine becomes cracked, and an intine comes out in the form of a small tube. That small tube is called as the promycelium. Now all the contents before this, in this zygospore, meiosis takes place. That means all the nuclei which are here in a zygospore becomes haploid in nature. Then all the nuclei with cytoplasm and food material get migrated into newly formed tube-like elongated hypa, which is called as the promycelium. Now this promycelium at its tips forms a small circular structure which is called as the vesicle. Now all the content, that means all the haploid nuclei which are here in this promycelium get migrated into this newly formed vesicle. Now this vesicle is get metamorphosed into another large structure, and that structure is called as the zygosporangium. Now it is developed at the tip of the promycelium. Now in the zygosporangium there are very large number of the meiospores, and at the base there is the columella. Now when these meiospores becomes mature, at that time the wall of this zygosporangium dries up and all the spores get dispersed with the help of wind, and now all of these meiospores get germinate into a new mycelium. But before this newly formed meiospores, there are the two views. That means according to one view, all the meiospores are of the plus strain, or all of the meiospores are of the minus strain, or according to another view, half of the meiospores are of the plus strain and half of the meiospores are of the minus strain. But they are formed there, they are called as the meiospores, and finally they get germinate into a new mycelium of the Rhizopus. Thank you.